Welcome to The Explainer. Today, we're going to dive deep into the absolutely incredible life story of a T cell. We're talking about one of the most important players in your immune system, and we're going to follow it from the moment it's born right through to its final mission.
So let me just start by asking you a question. Who are the expert assassins and, the elite commanders inside your own body? The ones that are trained with this unbelievable sensitivity to hunt down and destroy very specific threats, like viruses or even cancer cells.
Well, if you guessed T cells, you are spot on. They are the highly specialized elite operatives of what we call your adaptive immune system.
So here's our game plan. We're going to follow the truly heroic journey of a T cell. First, we'll meet these elite operatives. Then, we're going to watch them go through a seriously brutal boot camp. We'll follow them on their search for an enemy, see the super precise signals they need to get the call to arms, watch them build a whole army of specialists, and then, finally, we'll get into their lasting legacy of memory and ceasefire.
All right, let's get right into it and meet these guys. At the highest level, you can think of T cells as having two main jobs, and these jobs are carried out by two different classes of cell. And this is where it gets really interesting, this division of labor. On one side, you've got the CD8 plus T cells. We call them cytotoxic T lymphocytes, or CTLs for short. These are the frontline assassins, the ones that perform cytotoxicity. Their job is to literally find and kill your own cells that have been infected. On the other side, you've got the CD4 plus T cells. Think of these as the battlefield commanders. They don't do the killing themselves. Instead, they direct the whole operation by releasing these chemical messages called cytokines, which activate and call in backup from other immune cells.
But here's the thing. Before a T cell can do any of that cool stuff, it has to survive an intense, unbelievably brutal training program. And this all happens in a special little organ called the thymus.
This timeline lays out the whole journey. It all starts as a simple stem cell in your bone marrow. From there, it travels to the thymus, where it gets a new name, a thymocyte. Now, in the thymus, it develops its one-of-a-kind weapon, the T cell receptor, or TCR. And then it has to face two life-or-death exams. This process is so ridiculously tough that, and this is just wild, only about 2%, just 2%, actually survive to graduate and become what we call mature, naive T cells, ready to go on patrol.
Okay, so you've got to understand this two-step quality control because it is absolutely critical. The first test is positive selection. It basically asks, hey, can you even see our body's own ID cards? Those ID cards are the MHC molecules. If a T cell can't recognize them at all, it's totally useless. It can't get any signals, so it's eliminated through a process of programmed cell death called apoptosis. Then comes negative selection. This test asks, okay, you can see our ID cards, but do you react too strongly to them when they're showing our own stuff? If the answer is yes, that cell is a potential traitor, it's autoreactive, and it is way too dangerous to let out, so it's also eliminated. So, after our T cell somehow graduates from this insane boot camp, this brand new, naive T cell begins its life's work, a constant patrol through your lymph nodes, scanning, always scanning, for the one specific enemy threat it was built to see.
Now, T cells have a very particular way of seeing the world. They don't see whole viruses or whole bacteria. Nope, they see tiny little pieces of them, fragments called peptides. These peptides are the specific targets or antigens they're hunting for, and they're served up on these molecular platters on the surface of other cells. These platters are called major histocompatibility complex molecules, or MHC for short. And this whole process of showing the peptide on an MHC molecule, that's called antigen presentation.
This is just a brilliant system. It's how basically any cell in your body can raise the alarm when it's been hijacked from the inside. Let's say a virus gets in. Its proteins get chopped up by a cellular machine called the proteasome. Those little peptide fragments are then loaded onto MHC class I molecules inside the endoplasmic reticulum and then shoved out to the cell surface. This is, for all intents and purposes, a kill me signal, a flag that tells any passing CD8 plus killer T cell, I'm infected, take me out.
Now, the MHC class II pathway is a bit different. This one is used by the professional scouts of the immune system, the antigen-presenting cells, or APCs. Their job is to find threats that are outside our cells. They gobble them up, break them down into peptides, and then load those peptides onto MHC class II molecules. There's a really neat quality control step here. The slot on the MHC molecule is initially blocked by a placeholder called the CLIP peptide, which is a remnant of a larger molecule called the invariant chain. Once the real enemy peptide is ready, clip is removed, the enemy peptide is loaded, and the whole complex is put on the surface to tell the CD4 Plus commander T cells, hey, there's danger out there. We need a coordinated response now.
So our T cell is patrolling, scanning, searching. What happens when it finally finds its target? Well, you might think it just immediately goes on the attack, but it doesn't. And that's because there's a critical safety check, a system designed to prevent accidental activation and, total autoimmune chaos.
It all comes down to the two-signal hypothesis. Signal 1 is specificity. This is when the T cell's receptor physically locks onto its specific peptide MHC target. That's the what. I found my enemy. But that's not enough. To get fully activated, it needs signal 2. This is the danger signal. It happens when co-stimulatory molecules on the APC, like B7, bind to a receptor on the T cell called CD28. This second signal is the confirmation, the why. It tells the T cell that this enemy is part of a real, genuine threat.
And this two-signal system is an absolutely brilliant safety mechanism. If a T cell gets both signal 1 and signal 2, boom, full activation. It starts pumping out its own growth juice, a cytokine called interleukin-2, and starts cloning itself into a massive army of what we call effector cells. But if it only gets signal 1 and there's no danger signal, something totally different happens. The cell enters a state called anergy. It basically becomes unresponsive, a zombie cell. It has now learned that this target is harmless, which is how we avoid attacking our own healthy cells or, say, particles from the food we eat.
All right, so once it's fully activated with both signals, that single, lonely, naive T cell undergoes an incredible transformation. It starts dividing like crazy, proliferating, and then it differentiates into a huge army of specialized effector T cells, each one trained for a very specific job.
And this is where that quote from our source material really just hits the nail on the head. It says the CD4 Plus T cell truly is the commander of the adaptive immune response. It's the one giving all the orders that will shape the entire battle.
Just look at this table. It shows the amazing specialization of that CD4 Plus army. I mean, depending on the threat, that commander cell can become a different type of general. You've got Th1 cells, which are experts at fighting stuff inside our cells, like viruses. Then you have Th2 cells, which are geared up to fight bigger things like parasites. Th17 cells are specialists at recruiting other soldiers to fight off bacteria. And maybe most importantly, you have regulatory T cells, or Tregs. They're the peacekeepers. Their job is to suppress the immune response and shut everything down when the war is over. Each one is a unique commander giving unique orders for a totally unique fight.
Okay, now let's talk about the assassins, the activated CD8 Plus T cells. These guys become cytotoxic T lymphocytes, and the source material describes them perfectly. It calls them serial killers. They hunt down a target, they kill it, and then they just detach and move on to the next victim and the next and the next.
And the way they kill is just elegant. It's not some messy explosion. The CTL latches onto its target and delivers this lethal hit from its toxic granules. This hit has two key weapons. First, a protein called perforin literally punches holes in the target cell's membrane. This opens the door for the second weapon, called granzymes, to get inside. These granzymes are enzymes that activate the target cell's own self-destruct program, the caspase pathway, triggering that controlled death we call apoptosis. It's a clean, controlled demolition that makes sure the virus dies with the cell and doesn't cause a bunch of collateral damage.
Okay, so the infection is cleared, the war is won. Now what? The immune system can't just stay on high alert forever. That would be exhausting and damaging. It has to stand down and call a ceasefire.
So, after the fight, the vast majority of that T-cell army is no longer needed. They actually die off through apoptosis, and those trig cells we mentioned help calm everything down. But, and this is the amazing part, a small population of veteran cells survives. These survivors become long-lived memory T-cells. They can circulate in your body for years, even decades. And if that same enemy ever dares to show up again, these memory cells are ready to launch a response that is so much faster and so much stronger than the first time. This right here, this is the entire principle behind how vaccination works.
And let's just circle back for a second to this mind-blowing number, 2%. For every 100 T-cell candidates that enter the thymus for training, only 2 make it out alive. This incredible, extreme selectivity is what ensures your T-cell army is both a ruthlessly effective killing machine against invaders and, for the most part, incredibly safe for you.
When you look at the whole thing, it's just a masterclass in control, in specificity, in power. It's almost a perfect system. But that leaves us with one final, and pretty chilling question. What happens when this powerful, precise system makes a mistake? What are the consequences when the body's own expert assassins declare war on the very person they're supposed to protect?